Hey everyone, I'm going to take you guys through the protocol for the beta-galactosidase lab here. We are not doing the beta-galactosidase part 1 lab in class because of the formatting of this term and reduced lab time, but you guys will be doing beta-galactosidase part 2, wherein you get to do some experimental design around variables that you'd like to investigate related to this experiment. So we thought it would be helpful for you to see how the experiment works and how the equipment works in advance of trying to do that experimental design on your own. So the first thing I want to do is just give a very quick background on what it is we're actually doing in this lab. So I'll share screen here quickly and show you what we've got going on. So lactose is a disaccharide composed of glucose and galactose, and the enzyme beta-galactosidase cleaves lactose in half into separate monosaccharides, galactose and glucose. So we'll be using that enzyme beta-galactosidase to investigate enzyme activity with respect to a variety of different variables. But to give us a system where it's easy to measure enzyme activity, we'll actually be using a slightly different substrate called OMPG, which is very similar to lactose, except that instead of containing glucose as the second monosaccharide in the disaccharide, it contains a di called OMP. When beta-galactosidase cleaves the OMPG molecule separating galactose from the OMP di turns yellow, and that allows us an opportunity to see how yellow our reaction tube has become, and therefore how much enzyme activity has occurred. So that's the system that we're going to be using to investigate enzyme activity. And let me now stop the share and walk you through a little bit of exactly what we're going to do. The first thing that we're going to do is set up all of our test tubes. And let me show you what we're looking at here. Okay, so we have here, if you guys can see with my camera, a whole bunch of test tubes set up. And we're going to put a few different reaction components into those test tubes. We will have the substrate itself. These are the tubes of OMPG. That's that molecule I just showed you, which has galactose and the OMP di on it. So that'll be our substrate. We'll be adding that into the tubes. We also have over here on ice our working enzyme. This is the enzyme, the beta-galactosidase, which we will also be adding into the tubes. The first component that we'll actually be adding into the tubes, though, is a reaction buffer. So I'm going to take you over to the hood where the reaction buffer lives and show you that. Here under the hood, we have our reaction buffer. So that's where I'm going to get the buffer from, and I'm about to fill it up into the test tubes. So I'll set up the camera just here, so you get a view there under the hood, while I bring my test tube rack over and use a pipette to fill up each tube with the appropriate amount of buffer, which will be 4 milliliters. So here I have my test tube rack, and here I have the pipette that I'm going to use. You may have used these pipettes before. You can use your thumb on this scroll wheel here to pull fluid up into the pipette, okay? And then you can use this lever on the side to push and release the fluid down into the test tube. As you can see here, there are graduations on the side of the pipette here, which will give me a sense of how much volume I've actually gotten, okay? So without further ado, I'm going to move some buffer into my tubes. Great. At this point, I have put the buffer into each of the tubes that I need, and I will now move those tubes back to my station and bring you over there to show you the next step. Okay, in my next step here, I'm going to be adding into each of those tubes some of the substrate. I'll be adding 1 milliliter of substrate of a variety of different concentrations of the substrate into each of these tubes. Okay, I'll need to use a separate pipette for each of those because I don't want to cross-contaminate my substrate concentrations, and they are all different. Okay, so without further ado, I will add that substrate into each of my reaction tubes. As you can see here, I have on ice my OMPG substrates, and they are in a variety of concentrations. So I will just use these pipettes right here. The 1 milliliter marking is right there at the very top of the shaft here before the ball. And so I'll just go ahead and add the substrate in now. All right, excellent. I have added my substrate, the OMPG, in a variety of concentrations into each of the tubes that you can see here. The next thing I'm going to do is will be to add the working enzyme at 1 milliliter into each of these tubes. Now, the addition of the working enzyme is a time-sensitive step because as soon as I add the working enzyme, the reaction will begin, and therefore I want to have exactly the same amount of reaction time for each of these tubes. So what I will do is use a timer, and every 20 seconds I will add another 1 mil of working enzyme into the next tube. So when it comes time to stop the reactions, I can stop them on that same scale of time. So I'll turn my stopwatch on here, and the moment I add my working enzyme into the first tube, I'll start it. And then again, every 20 seconds or every 30 seconds, whenever I just need to establish exactly how long I'm going to give it, I will then go into the next tube. The working enzyme is going to be the same for each of the tubes, so I don't actually need to use a different pipette to put it into each of the tubes, so long as I don't dip my pipette tip actually into the fluid in the tube itself. Okay, so here we go with my working enzyme. Get my 1 milliliter here. Okay. And as soon as I put that into tube 1, I'm going to start my timer. As it runs, I'm going to just pipette up and down to mix in that reaction tube so it's thoroughly mixed, and then prepare myself to add into my next tube at the 20-second mark. Now that I've put the working enzyme into each of the tubes that I'm going to be using, I will simply wait 10 minutes for the experiment to run, for the enzyme to work, and then I will show you how to stop the reaction and then collect the data from this experiment. Okay, I'm just about at the end of my 10-minute timer, so I'm going to get ready to stop these reactions. To do that, I'm going to be using this stop solution right here, and I will be using this red pipette, red for stop, to dispense the 1.5 milliliters of stop solution into each of these tubes at the same 20-second intervals that I added the enzyme with. So I will start by drawing up the stop solution, and then when the time comes, I will dispense 1.5 milliliters at a time. As you can see, looking at these reaction tubes, they have a little bit of a yellow tint to the solution, which again is an indication to us of how much of the reaction has proceeded. Now I'm going to be using these cuvettes to transfer the reaction solutions into and measure the absorbance, which essentially quantifies exactly how yellow each tube is. As you look at these little cuvettes, you'll see that two sides are clear and two sides are ridged. When we line these up in the spectrophotometer, which is the device that will measure absorbance for us, we're going to need to make sure that the clear sides are lined up with the direction that the light shoots through. I'll show you that in a second. We also want to make sure that as we handle these cuvettes, we're only handling them by the ridge sides, so we're not getting a lot of fingerprints on the clear sides, which would affect the amount of light that can pass through. So what I will do is fill a cuvette for each of these samples. Now I'm going to start with this tube over here, which is actually a tube to which I added no enzyme. It's my blank. This will serve as my control because it will tell me exactly how much absorbance I expect when I know that no reaction has taken place. So I will use my pipette in that tube to fill up my cuvette here. Specific volume, not super important, just needs to be probably about 3/4 of the way filled up. Then I can put my cap onto it and I can head over to the spectrophotometer. I'll take you over here to the spectrophotometer now. Okay, this is the spectrophotometer. It should be set up. You can kind of see here. With 420 nanometers set as the absorbance, as the wavelength for measuring absorbance. And then I am going to open up right here and try to give you a look inside the spectrophotometer here. There is a little place for me to slide my cuvette. The light shines front and back like this. So I need to make sure that my ridge sides are oriented sideways, my clear sides are oriented front to back. I slide my cuvette in, I close the lid, and for this first one, I'm going to hit 0 absorbance / 100% transmission. This is essentially my tare button to say however much absorbance you saw in this sample, count that as 0 absorbance, because this was the one in which I know that no reaction took place. Okay, now I'm going to take that sample out, set it aside, and move on to my next sample. All right, there we go. At this point, I have collected the data from all of my samples, and as you've been able to see, I actually got an increasing amount of absorbance as I went through, which is a result of increasing amounts of yellow. I'm going to try to show you the tubes over here. You can probably actually see just on visual inspection that there's an increasing amount of yellow as I go from left to right, which is just an indication that I've had more progress of the reaction, because remember that the reaction progress is indicated by the cleavage of galactose from the OMP dye, which results in that yellow color in the solution. At this point, it's a matter of cleanup, and I'm going to dump each of these little cuvettes into the waste beaker under the hood, and I'll be all set to go. I just want to show you quickly what my data looks like here, so you can get a sense of what we're looking at. And there's the data that I've quickly collected on this. You can see here that I had a blank setup, which I had set as a 0 absorbance, and the rest of these are my actual absorbance values from my various OMPG concentrations. From here, we'd be able to proceed with our analysis, and you, in the lab that you'll conduct, will be able to decide what variable you want to adjust and investigate its impact on OMPG cleavage by beta-galactosidase, and therefore its impact on enzymatic catalysis of reactions. So that's all for now. Hopefully this gives you a sense of how the lab runs, how the equipment works, and will put you in a position to explore a variable of your choosing in a couple of weeks.